All right, Dr. Crosswhite here. Let's go ahead and finish up the remaining information as it relates to the digestive system. So this lecture is going to pick up with, learning objective number 15, where we'll begin by discussing some general functions of the stomach. That'll lead us into processes and control pathways of the gastric phase, which will then take us into some specifics on the gastric secretions, the enzymes, and their major action. And then we'll go into comparing motility and especially digestion in the large and small intestine. A vast majority of this information will relate to the small intestine. We'll cover very little on the large. And then we'll discuss some accessory organs, including or part of the liver, the hepatic portal system, but especially the gallbladder, the pancreas, and the liver. And then we'll transition into the actual mechanisms for how many of these nutrients are actually absorbed in the enterocytes. So we'll get into some specific information for absorbing carbs, proteins, and fats or lipids. And then that will basically finish us up. We'll very quickly go over hormonal control, and that's really just a review slide. It's not new information at that point. We'll go into a little bit of the large intestine, and then just real basic information on vomiting and diarrhea. Okay. Okay, so we've gone over the cephalic phase. Not a lot of actual digestion occurs there, as you learned. So now let's shift gears and talk about the gastric phase and the intestinal phases. So 3 general purposes or functions of the stomach. So you may not think of it, but the stomach is a temporary storage organ. Okay, so it helps store our food. What you're probably more familiar with is the concept of the actual digestion. So we're talking mechanical or chemical digestion here. And there's going to be lipid and protein digestion that occurs, not carbohydrates. We'll cover why that is here in a few moments. And then we'll go into the specifics of acids, enzymes, and some of these paracrine molecules, and especially hormones. Third function of the stomach, as I've mentioned before, that stomach acid is a defense against swallowed pathogens. So hopefully if you take in some bacteria or viruses, that the very strong hydrochloric acid will be enough to kill any of those pathogens. Okay, so one thing that's covered very briefly here, it says digestive activity in the stomach begins with the long vagal reflex of the cephalic phase. Okay, so we talked about these feed-forward mechanisms previously and how the parasympathetic nervous system is involved. So if you remember that many parasympathetic fibers run through the vagus nerve, okay, that's what we mean by a vagal reflex. So the sight, the smell of food, that's going to be initiated through a vagal reflex that starts preparing the stomach to receive the food that you're about to take in. Okay. So a little bit more on the stomach storage. Basically, stomach is holding food so that your small intestine can properly absorb the nutrients it needs to. Okay. Most of the storage actually occurs in the fundus. Okay, so that's the anterior or top portion of your stomach. While the lower portion of your stomach, the body and the antrum, that's where most of the mechanical and especially any chemical digestion is going to occur. Now, I mentioned that storage in the stomach holding food so that proper absorption can take place. What will result if your stomach does not store that food for a proper amount of time before it's being released into the small intestine is something called that dumping syndrome. And that's when people eat too fast or when their meals are high in lipid content. It's going to trigger this. As well as individuals who've had surgical removal of part of their stomach and their stomach is just much smaller, they can suffer from the chronic dumping syndrome. Okay. Another term that I may or may not have used in class, chyme. Really what chyme is, and I'll cover it again here in a few slides, but chyme is the mixture of food and then your stomach. So it's food plus like the acid and enzymes that are released from your stomach. So don't get tripped up on the term chyme. But stomach really needs to control that chyme and the release of it into the small intestine. Okay, it's only small amounts at a time, maybe around the order of 40, 50 mils, maybe 100 mils of chyme at a time. It doesn't release all the stomach contents into the small intestine all at once. Okay, so just a reminder on some of the anatomy as it relates to the stomach. We've talked about the fundus, the body, and the antrum. And what I just mentioned right here at the pylorus, and especially there's a sphincter here, the pyloric sphincter, that's what controls the rate of the stomach contents into the small intestine. And then just as a reminder, remember that especially in the body and the antrum of the stomach, it's really, the mucosal layer is really folded and increases that surface area. Okay, so what we're going to talk about is what's happening in these cells of the crypts. If that's a term that we'll use, the gastric crypts. Okay, so we've talked a little bit about mucus and how the epithelium needs that mucosal, that layer of mucus to help protect it. Okay, but what we haven't really talked about are, in addition to that mucus lining, in the gastric pits and the cells that are present there that release these enzymes and or acid. Okay, and there's multiple cell types that all secrete different substances, and they're going to secrete acid, various enzymes, potentially hormones, and definitely paracrines. Okay, and we're going to go over that. So this is just kind of a brief introduction, but stomach has a lot of secretory processes that occur. So here's a diagram. So we're looking in the body of the stomach, and we've zoomed in, and this would be the lining of the mucosal layer, so the rugae up here, and now we're looking into one of these crypts or gastric glands. Same, I use the terms interchangeably. Okay. And I do expect you to know what the cell types are, what they're secreting, and what the function and stimulus for release is. Okay? But you'll see that there's a lot of patterns to this, so it's not as much information as you probably are thinking right now. So we'll start at the top. So the cells that actually line the exterior or the mucosal layer out here in the rugae, the mucosal or mucus surface cells, they're going to be the ones responsible for secreting mucus. Okay? And the function of that, as we've discussed before, physical barrier between the lumen and the epithelium. It protects the epithelial cells. A little further down from the surface cells, we've got these neck cells. So they're here in the neck of the gland. Okay? And they don't secrete mucus, but what they do secrete is bicarbonate, okay, or bicarbonate. And then, as we discussed in class earlier, bicarbonate is a really good buffer against acids. Okay, so let's see. We'll get into. So one thing I want to emphasize right now is that they're not constantly secreting bicarbonate, because you don't want so much carbonate that it neutralizes all the hydrochloric acid. Okay, moving a little farther down into these glands, we encounter our first group, cells called parietal cells. Okay, so parietal cells, they're the ones that actually produce the gastric acid or the hydrochloric acid. Okay, and we need the hydrochloric acid. As I've mentioned, it'll help kill pathogens, bacteria, potentially viruses. But this acid, it will activate an enzyme called pepsinogen, and it'll convert pepsinogen into pepsin, and that helps break down proteins. Okay, and we'll cover this. We'll re-encounter this here in the near future. Okay. Stimulus for the release of acid is the hormone gastrin, which we'll talk about here in a second, the presence of histamine, which we'll talk about here in a second, but also acetylcholine. So think about those parasympathetic reflexes or processes, and that when parasympathetic activity increases, that acetylcholine is going to help stimulate the production of acid. The second thing that the parietal cells release is something called intrinsic factor. And we'll cover this later in the chapter later in the semester when we go over vitamin absorption. But really, intrinsic factor is very specific. We need it so that our body can absorb vitamin B12. Okay, I'm not going to focus on it a lot right now, but we'll encounter it in a few weeks. The next group of cells are these enterochromaffin or ECL cells. ECL for short. So these ECL cells, they secrete histamine. So you might associate histamine with immune functions, immune reactions, but histamine is a very important paracrine. Okay? And histamine is going to help cause or stimulate the release of the gastric acid. So histamine acts on those parietal cells so that we can produce stomach acid. And again, what stimulates its release? Acetylcholine, okay, as well as gastrin. The next group of cells are chief cells. And just like the parietal cells, they release a couple of different substances. One I've mentioned before. So chief cells are where the pepsinogen is produced. Pepsinogen is an inactive enzyme, okay? And it's pepsinogen, when it's activated into pepsin, it will start breaking down proteins. So this is where we get into the first example of some chemical digestion that's occurring in the stomach as opposed to mechanical digestion. And just like the other cells above, acetylcholine and acid are a stimulus for its release. Okay. And then the last two cells, we've got the D cells. D cells, what's going on with the D cells is that they release a substance called somatostatin. And somatostatin is like the brakes for the stomach. Let's say food has entered the stomach, it's been mechanically and somewhat chemically digested, we've produced a lot of stomach acid. Well, at that point, when the stomach acid builds up to a certain level, the D cells release somatostatin, and it really kind of calms this whole process down. So it's an inhibitory, somatostatin is an inhibitory chemical. Okay. So it inhibits that gastric acid secretion, so you can imagine that its target are the parietal cells, and it's stimulated by the abundance of acid in the stomach. G cells, these are the ones that actually produce gastrin that I've been talking about this entire time. So gastrin, one of those hormones, stimulates, again, gastric acid secretion. So gastrin acts on parietal cells, and just like the others above it, acetylcholine is one of the major stimuli for release, okay? As well as the presence of protein in the stomach, peptides and amino acids. Okay, so those are the secretory cells of the gastric mucosa. You're probably starting to appreciate the complexity of the stomach and all that's going on when you take in food. So just a little bit more on some of the secretion. So gastrin obviously is a big one. A lot of things target or produce, or gastrin obviously has many different targets. Okay? So one thing I didn't really emphasize previously is it's acting as both a paracrine and a hormone. Okay? Because when those G cells release gastrin, they're releasing it into the blood supply, but they're also releasing it to the nearby cells in those glands. So that's how it's acting as both a paracrine and a hormone. Okay? And like I have already established, that it's released in response to what I mentioned was a parasympathetic or cephalic reflex, but as well as enteric nervous system reflexes. All right, a little more on hydrochloric acid itself, or just acid. Those parietal cells, on average, an adult will produce around 1 to 3 liters of hydrochloric acid per day. That's a lot of a very strong acid. And as a result, at any given time, your stomach has, the environment in your stomach has a pH, the fluid in there has a pH as low as 1. That's extremely acidic. It's one of the most acidic acids, acidic environments that I know of. Okay, and the acid in the stomach has many different functions. Okay, it stimulates the release of pepsin. Okay, and really it's the release of pepsinogen. So you need hydrochloric acid to stimulate this release, but it also, the acid will cleave pepsinogen into the active form of pepsin so it can start breaking down proteins. In addition to breaking down proteins with pepsin, the stomach, or the acid, will directly denature some of the proteins. Okay, so pepsin is working, acid is working, all on proteins. And when the acid builds up to a high enough level, it will trigger that inhibitory chemical somatostatin. Okay? And we've already covered how it can kill bacteria and other microorganisms. Now here's one thing that I haven't mentioned so far, and we talked about it in the cephalic phase, but it was the role of salivary amylase in breaking down carbohydrates. As soon as that salivary amylase hits the stomach contents, it gets inactivated by the acid. So there is no carb digestion in the stomach. Okay, moving on. Let's talk a little bit more about acid secretion and how that happens. We've got a parietal cell here. And one thing, I don't expect you to know the mechanisms of this in detail, but I just want you to appreciate or realize that when we say the parietal cells produce acid, they can't produce HCl here on the interior of the cell. That is not possible because as soon as HCl is produced, it would start breaking down that cell itself. So what the parietal cells do is, through this bicarbonate exchange pump here that we've talked about in red blood cells, or that you've encountered in red blood cells, the chloride exchangers, they bring chloride in and they pump chloride out into the lumen of the stomach. At the same time, you have a hydrogen potassium pump, okay, or hydrogen potassium ATPase that is pumping hydrogen out in this direction. So then, once hydrogen and chloride are out here, that's when you have the formation of hydrochloric acid. So it doesn't happen within the parietal cell itself, happens out here in the lumen of the stomach. And then the other thing, many of you have probably taken one of these, but the purple pills, aka, they're proton pump inhibitors, so Nexium, the other names are escaping me at the moment, but they're those over-the-counter medications to reduce heartburn. Most of them are targeting this hydrogen potassium ATPase. I can't remember exactly how they either degrade it or they cause it to be internalized into the parietal cell, but the point is they reduce HCl production in the parietal cells. Okay, just real quick, since we've talked about this previously, this is kind of giving you a generalization of how the gastric juice and the mucosal layer and the gastric mucosal cells are all operating. But out here in the lumen, in the presence of the hydrochloric acid in your food, this is where all that mechanical and some chemical digestion is occurring. Then you have that mucosal layer that's a physical barrier. Any acid that happens to penetrate that mucosal layer hopefully is neutralized by bicarbonate. Okay? but some things can get through. If acid gets through, then it has the potential to actually destroy those mucosal cells, and that's where you can get an ulcer and some bleeding. Okay. All right, so we're still talking the gastric phase here and some of those secretions. I've gone over these. I've gone over one of them a little bit, but we'll talk about a second one. Okay. Pepsin, that initial protein digestion, not all of it, but it starts the process of breaking down proteins. Pepsin itself is particularly effective against collagen, which is a form of connective tissue. So it breaks down collagen, also breaks down protein. So it's really effective against meat because meat, especially red meat, is rich in collagen. Okay. Gastric lipase, which we haven't talked about it being released from. There is a little bit of lipase that's being produced in the stomach, but really, and it's starting that fat digestion, but really there's, again, minimal lipid digestion that's occurring in the stomach. Most of it occurs in the small intestine. Okay. Again, this is just information that we went through previously, so nothing new here. Histamine released by those ECL cells. Intrinsic factor is needed to absorb vitamin B12, and somatostatin is really that negative feedback or that inhibitor of the secretion in the stomach. Okay, so that's it on the gastric phase. Now we're going to shift gears and talk about what happens when that chyme is moving from the stomach and into the small intestine. So chyme, it's being released in a controlled manner into the small intestine. It's not all at once. It's small amounts over a period of time. Okay, and we've talked some about chemical digestion. There's been a little bit. Okay, but here in the small intestine is where we encounter a lot of chemical digestion. Okay, and as I mentioned before, motility is really important to control that chyme and how it's entering the small intestine, because the important point here is that our small intestine needs time so that we can increase the amount of nutrients that we're absorbing by the enterocytes, which we'll get into here in a little bit. Also, just some kind of big picture information here. Between food and fluid that you take in, as well as all these secretions, salivary, so saliva, stomach secretions, secretions by the accessory organs in your small intestine, it all comes out to, roughly 9 liters of food and fluid per day. Okay, over half of that is coming directly from the food you take in, the fluid, and salivary or stomach secretions. And then about a third of it is due to the presence, or due to the secretions from the liver, the pancreas, and the intestine, and the gallbladder as well, but the gallbladder is a minimal amount. So it's a lot of stuff. Okay. Before we get into some of the nitty-gritty of how these nutrients are being absorbed, we need to talk about some anatomy of the small intestine. So just as in the stomach, and you had those rugae, I should say, that were a modified epithelium, we have the same thing here in the small intestine. Instead of rugae, we've got the villi. And if we were to zoom in, so this is just, increased surface area of the small intestine. If we were to zoom in to the border of the villi, what we'd find are individual cells, and they're called enterocytes. The enterocytes have additional increased surface area that we refer to as the microvilli. Okay, so lots of surface area here in the small intestine. And the microvilli, important point to realize right now, absorption does not happen at the level of the entire villi, or villus for singular. Absorption happens here at the level of the microvilli, but we'll get into that later. Okay. The other thing I want to point out, each villi or villus, okay, has a blood supply. So has arterial blood supply, has a venous blood supply, because that's where the nutrients are eventually going to be absorbed into once they pass through the enterocyte. And they also have a structure that is referred to as a lacteal. That's just a fancy term for part of the lymphatic circulation. And when we discuss the digestion and absorption of fats, we'll come back to these lacteals. Okay. But this is kind of an important point right here that we'll still have time to talk about. But proteins and carbs are going to enter the capillary system or the circulatory system, whereas the fats are going to enter the lymphatic circulation. Okay. And then we also have the luminal crypts here that there are more secretions. Okay, so learned about villi and microvilli. The other thing, before we get to the details, I want to talk about accessory organs of the digestive system. So liver, gallbladder, and pancreas for the most part. Now, if you think about when you take in a meal and you're going to absorb you're going to absorb these nutrients, okay? They're all going into the carbs and proteins. They're going into the capillary system of your small intestine, okay? But your digestive system actually has a portal system, okay? So when you are taking in all the nutrients from your food and they're being absorbed across those enterocytes, it doesn't go directly into your circulatory system. Instead, what happens is all those nutrients get funneled into a portal vein that goes directly to your liver. And the reason is the liver is really efficient and really good at metabolizing drugs and toxins. You can think of the liver, one of its biggest functions is it's a filter. It filters the food that you take in to try to reduce your exposure to harmful substances. Okay, this is really good for our body, but there's also a downside to this. Because the liver is so good at metabolizing, it leaves the nutrients alone, but it will metabolize drugs, toxins, etc. And because of that, the liver is actually a big hurdle when it comes to designing drugs. Any drugs that are delivered orally, they're going to hit the liver, and drug companies have to think about, well, how's the liver going to metabolize that? How much of is it going to get through the liver, and how much of the drug will be delivered? For an example, acetaminophen, aka Tylenol, at least 90% is metabolized by the liver. And I don't know the number off the top of my head, but I think it's probably somewhere between 2 and 4% of a single dose of acetaminophen is what actually makes it into your bloodstream. Okay, so liver is really important. Helps filter the blood and the nutrients from everything that you're taking in. The other thing is, let's talk about a little more on the liver and the gallbladder especially. So it's kind of a funky looking photo over here. We've got the liver up here. We've got your stomach here. So right here would be the pyloric sphincter, and right down here would be the beginning of your small intestine, the duodenum. Okay. So you also have this other accessory organ right here. This is your gallbladder. Okay. So thing to point out, bile, which we'll talk about here in a second, is actually, so bile is made in the liver. Okay? It's a common misconception that the gallbladder produces bile. That's false. The liver makes the bile, gallbladder stores it. Okay? And then there's a duct that connects the bile that's being produced in your liver and the bile that's stored in the gallbladder. It's called a common bile duct, but this is shown right here, and it connects just at the beginning of that small intestine, the duodenum. And that's because as nutrients are hitting your small intestine, bile will be released into the small intestine. We'll talk about the role of bile here in a little bit. Okay, and just as a reference, what controls the release of bile into the small intestine is this sphincter called the sphincter of Oddi. Okay, so that's a little bit of background on gallbladder. I don't expect you to know this, but all this being said, the liver is vitally important for not only digestion, but many, many different metabolic functions in our body. So take some time to just go through this diagram here. It's a little confusing at first, but it's basically kind of arranged it by what you're bringing into your body from the GI tract, what's absorbed or what's being released from peripheral tissues, so your cells. It's how it's leading to the liver, and then some of those roles that the liver is providing. Okay. All right. Still some surface or not surface, but some information before we actually get into the absorption of the nutrients. Okay. In addition, so we're talking secretions now. We're looking at the crypts of the small intestine. There are these structures called goblet cells that I haven't shown, or I didn't show previously. But just like the stomach, we need some mucus there to help lubricate, to help protect those epithelial cells, especially in the duodenum because of the presence of gastric acid. Okay. And I just touched on it, but the liver is where our body makes bile. So bile is actually a combination of bile salts and pigments and cholesterol. And the important point about bile is that we absolutely need it in order to digest fats or lipids. Okay. When we talk about secretions and accessory organs, we can't ignore the pancreas. Most of you probably associate it with insulin and and glucose and insulin secretion. But in nutrition, we're going to go a little beyond that. Okay, so what I want you to know about the pancreas is, yes, it has endocrine functions, as you've learned previously in regards to insulin, but pancreas also has a lot of exocrine secretions as well. Okay, so insulin released by beta cells, glucagon released by the alpha cells, but those are hormones. They go into the bloodstream, so that's why it's an endocrine secretion. However, pancreas also releases a lot of digestive enzymes, and because of that, it has exocrine functions. Okay, it's more than just bicarbonate, but we'll go into that. So in addition to bicarbonate, they're digestive enzymes. They're collectively called zymogens. It just means an inactive form of an enzyme, and we'll go over that a little bit when we talk about the digestion of proteins. Okay, so here is a view of the pancreas. So that view that I showed earlier, you would have had your liver and your gallbladder up here, and your stomach would be sitting in front of the pancreas actually. But notice it's at that same, it's in that same general area. We've got the beginning of the stomach would be here. We've got the beginning of the small intestine, the duodenum. We've got the sphincter of Oddi right here, and now we've got the pancreas, and we need to talk about how some of those exocrine secretions make it into the small intestine. Okay, so if we're talking hormones and the endocrine secretions, aka insulin and glucagon, they are being released into capillaries. There are a lot of capillaries, aka blood supply, in the pancreas. Those alpha and beta cells, they produce their hormones, they go into the circulatory system. That's different for the enzymes, for the digestive enzymes, and especially bicarbonate. The rest of the pancreas cells, they're called acinar cells, okay, and they are producing those enzymes, those chemicals, and they're feeding them into this pancreatic duct. Okay, so all those enzymes are being produced, and they're making their way through the pancreatic duct, which is shown here. So those enzymes travel through here, and then they're released into the small intestine. So bicarbonate, pancreatic lipase, pancreatic amylase as well. Okay, so A little bit more on zymogens that I mentioned earlier. So what we've got here is that pancreatic duct that I just went over, and those secretions, those inactive zymogens. All I really want you guys to know is that the reason they're inactive is because we wouldn't want them being active while they're in the pancreas, or in the pancreatic duct. So because that would defeat the purpose of having them. So instead they're inactive, and as soon as they hit the small intestine, they're activated. Gastric acid or hydrochloric acid will help with that. The enterocytes produce, what I should say is that the enterocytes produce enzymes that cleave trypsinogen into trypsin, and trypsin is the one that is actually going to cleave these zymogens. And I know there's a lot of fancy words here, but look at the end of them. Peptidase, lipase. So what all this means, okay, lipase, think fats, peptidase, think proteins. So these are the enzymes that start chemically, breaking down proteins and fats. All right, so we've established there's a lot of digestion, a lot of enzymes going on. Okay. Now we need to talk about how they're actually going to be absorbed. Okay. So after these nutrients are broken down from a larger size macromolecules into smaller micromolecules, that's the point where we can begin absorbing them or bringing them into our enterocytes. So we're going to start, let me back up here. So we're going to start with, when we talk about absorption, we're going to have to go through all three. We're going to start with lipids, and then we'll talk about how carbs are absorbed, and then we'll finish with proteins. Okay? So now we're going to incorporate a lot of these enzymes and hormones to accomplish actually bringing these nutrients into our enterocytes and into our body so that the rest of the cells can actually use them. Okay? So we've covered bile and it being produced by the liver and the gallbladder. So why bile is important for fat digestion, or absorption I should say, is when it's released in the small intestine, most fats at this point are present in these big lipid droplets. That's not a form that our cells can actually absorb. So the bile starts coating the lipids. Okay? And what it does, when it starts coating the lipids through mechanical digestion, the small intestine contracting, relaxing, it's going to break these large lipid droplets into smaller forms that we then can start absorbing the lipids into our cells. Okay? Another thing is that I think the next diagram is a little more straightforward, but one thing I want to emphasize before I move on, it's not real obvious over here, but the bile allows for the lipase and lipase and colipase, basically enzymes, to help break the triglycerides, so those are dietary fats, into free fatty acids and monoglycerides. This is what we can absorb. Our cells can't absorb triglycerides themselves. Okay, so what happens is you've taken in a meal, it contains fat, you've got bile salts from the gallbladder and the liver, and then through mechanical digestion of the small intestine, it breaks these large lipid droplets starts emulsifying them, taking them into smaller forms. They eventually get broken down into these very small lipid droplets that we call micelles. And the micelles, when they're present, when they come in contact with the microvilli of the enterocytes, that's when they can release the cholesterol and the free fatty acids and the monoglycerides. That's the point where they can actually get absorbed across the microvilli. Okay? And then what happens in the cell, this is a little weird. Digestion and absorption of fats is pretty complicated compared to carbs and proteins. Okay, so large droplets here, break them into smaller components. They get absorbed. Now here's where it's kind of weird. The enterocytes, they actually repackage those lipids into a substance called a chylomicron. Okay? And lipids, hopefully you know, is not soluble in water. So what happens, these chylomicrons, when they get released from the enterocytes, because the capillary system or our bloodstream is mostly water, we can't put lipids, very many lipids in our bloodstream. So instead, they are absorbed into the lacteals. Okay, so if you need a reminder of what the lacteals are, it's that lymphatic circulation. It's part of the villi. So you can go back to that diagram to see the presence of the lacteals in the villi. Okay? And we're not going to move beyond here in terms of what's happening to fat absorption and what goes on in the lacteals. We will cover that whenever we get to Chapter 5, I believe, when we go into more depths about lipids. Okay? So that's how digestion and absorption of lipids occurs. Carbohydrate digestion and absorption, much more straightforward, much more simple. Much of this information you should ring a bell. Okay, so big story here: we can't absorb large carbohydrates such as starch and glycogen. Okay, so the amylase that we've talked about, salivary amylase, pancreatic amylase, starts breaking those large carbohydrates into smaller units, usually in the form of 2 or 1, so a disaccharide or a monosaccharide. I don't expect you to know really the structure or the details here, but what I want you to focus in on is when we get the carbohydrates into the disaccharide or the size of a disaccharide or a monosaccharide is how they're being absorbed across the enterocytes. So we've got small intestine here, enterocytes. Now we've got the capillary network of the villi. When it comes to carbohydrates, glucose and galactose, which we'll cover in the next chapter, chapter 4, glucose and galactose are absorbed across the enterocytes through a sodium transporter. Once they're inside the enterocytes, then they are transported into the bloodstream. Okay? The other carbohydrate, fructose, it also enters the enterocytes, but on a different transport mechanism. They enter on GLUT5, they enter the apical membrane on the GLUT5 transporter, and they exit via the GLUT2. So here's one thing I want to pose right now. I'm not going to answer it, but I want to discuss it when we come to class, when we meet up in class, is how this process of bringing carbohydrates into these enterocytes, how that differs from the absorption of glucose that you've learned previously for most cells in our body. The enterocytes are special. They're unique compared to the other cells in terms of how they absorb glucose. So think about that and come to class next time we meet with ideas on why they're special or the details of that. Okay, that's digestion and absorption of carbs. Rounding this out, let's talk about proteins. Okay, again, a lot of information here. I'm not expecting you to know the details. We're just going over kind of the generalities. Again, we can't absorb large proteins or protein polymers. We have to break those proteins down into smaller fragments that we refer to as peptides, okay? Short chains of amino acids. They can be in the form of di or 2, tri, 3, so dipeptides, tripeptides. They're absorbed, those enterocytes absorb them through a transporter called pepT1, shown right here. Okay, don't expect you to know the details of that, or all I expect you to know is they're absorbing the protein through the use of a hydrogen transporter. Okay, we can also take in individual amino acids themselves, and then some smaller peptide fragments we can actually, those enterocytes can take in through the through transcytosis. So they can endocytize them here on the apical membrane. They can exocytose them on the basal membrane. But proteins, just like carbs, soluble in water, so they get put into the circulatory system, aka the bloodstream. Okay. I don't expect you to know endo or exopeptidases, but those are just enzymes that help break the large proteins down into individual amino acids or short peptides. Okay. Last couple things on the intestinal phase is just recognizing that there's more than just these 3 macronutrients that are being digested and absorbed. Okay? There's a lot of sodium absorption that will dictate or influence how water is absorbed. We have time in the rest of the semester to talk about that. There are vitamins and minerals that are being absorbed, as well as iron and calcium. These all have specific mechanisms that we'll cover throughout the semester. All right, so we've covered gastric phase, lots of details on the intestinal phase. This is really just kind of an overview for how these 2 phases interact and how they work cooperatively in order to promote proper digestion and absorption. I'm not going to go through the details of this slide right now for the sake of time, but I do just want you to pay attention to the diagram up here and just start at the top and work your way through and look at how the feedback occurs, the negative feedback for that most part, and then how these nutrients and solutions, what the presence of them mean in the small intestine and the results that happen, okay? And we can go over that when we come back in class. Right, so we're still on the intestinal phase, but really what I haven't talked about is the large intestine, okay? Really surface level information here. We'll have time to talk about large intestine more in the future. So I'm not asking you to know the depths of the anatomy, but I think at this point you should be familiar with kind of the generalities that there's, after the nutrients process or go through the small intestine, once it reaches the ileum, the ileum connects to the large intestine via the ileocecal valve. Okay, there's another sphincter here. But once the non-absorbed nutrients or the waste reaches the large intestine, has to go through the ascending colon, then it moves through the transverse colon, and then goes to the descending colon, and then you have the rectum. And zooming in on the rectum, you have 2 sphincters, internal and external. Okay, so basic anatomy of the large intestine. We're not even going into the depths of the role of the large intestine, just one slide on it. Some of the major functions of the large intestine, it's really concentrating that way so you can excrete it properly. Water and hydration status is really important for a good proper functioning of the large intestine. Okay, so if you're dehydrated, you're going to have a harder time with excretion. Motility is really important in the large intestine. Something called a mass movement. This is what projects the bolus or the waste through the large intestine, and when that bolus enters the rectum, the pressure of it, the presence of it there, stretch receptors is what's triggering the need, the brain to sense that you need to initiate defecation. Okay, and that is a reflex. It's a spinal reflex. So there's stretch receptors in the rectum. They synapse onto the spinal cord, and then the spinal cord will initiate the defecation reflex. You have obviously you have some control over this. So the external sphincter, not the internal one, but the external is under voluntary control. So you can override this defecation reflex through the input of the central nervous system, but that's to a certain degree. We've all experienced those situations where you need to go to the bathroom and you're holding off for as long as you can, but that desire, that urge becomes greater and greater and greater. Okay. The other big function of the large intestine that we won't go over right now is that it is really critical for the digestion of complex carbohydrates. Okay, not the simple carbohydrates, glucose, galactose, and fructose, but fiber and other complex carbs, we'll cover that later in the chapter. Okay. All right, that pretty much finishes up this chapter. Just one slide on the immune function, or a couple slides, I should say. And I'm really not even going into the depths, but throughout your small intestine, large intestine, you have a decent amount of immune cells that are present. You also have the release of cytokines. They attract immune cells to try to fend off any potential foreign invaders. They can trigger inflammatory responses, but we're really not, that's really all the level I want you to know at this point. And then you may not think of it, but vomiting and diarrhea, these are protective responses, not reflexes. Vomiting is a reflex. Diarrhea is not. Vomiting, aka reverse peristalsis, can be initiated by many different stimuli, but in class we can talk about why that's considered a protective response and the evolutionary reasons that we have the ability to vomit. Not all organisms, not all mammals have the ability to vomit. Okay. Diarrhea, we've all experienced this in response to many different factors. One of the biggest one, altered motility, so when motility is off, or infection from a foreign invader. All right, that finishes up this chapter. That's a lot of information, a lot for you to go through, but take some notes, make note of what questions you have, what you want to talk about when the next time we meet in class. Again, here are the learning objectives to emphasize, and I'll see you in class.